Okay, so this lecture is going to focus on cells, kind of doing a tour of the cell and the organelles within them, especially as they relate to plants. Cells were first discovered with the invention of the microscope in the 1600s, which really opened the world of things that are really small, right? Microscopic things. And what scientists soon found after looking through lots of tissues was that they all had cells. So some of the microscopes that we use today are the compound light microscope and dissecting scope, which also has a compound light function to it, but is just has a little broader range of view. We have some more some microscopes that can see things at a very, very small level or have a very, very high resolution. Those are the scanning electron microscope and the transmission electron microscope. So cell theory was first proposed by Schleiden and Schwann. Again, as they started looking through all these living organisms, they found that each of them had cells. The cells were different, but that they all had cells. And so the theory was that all living things are made of cells. New cells come from pre-existing cells, so they will split and form new cells. And they are the fundamental unit of living things. So the structure of cells breaks down into a bunch of different parts, each of them with different evolutionary origins, including infolding of the plasma membrane or endosymbiosis, which we'll discuss in a minute. But we'll go over these each individually. So we'll start, and we have this figure in the back. It's from your book. There's also this figure, which is where they actually stain different parts. So they have a different color, but this is a microscopic image as well, with some of the organelles, including the plasma membrane and cell wall. You have nucleus, ribosomes, mitochondria, and the big green chloroplast, and central vacuole, which are specific to plants. So the cell wall, so all plants have cell walls. Other types of organisms also have cell walls, but the cell wall of plants is different in that it is made of cellulose. Okay. It also has other structural components, including hemicellulose, pectin, glucoproteins, and lignin. There is a structure within the forming of a cell wall called the middle lamella. It is laid down first and is shared by two adjacent cells. It has basically a layer of pectin. And then the primary wall is laid down on both sides of the lamella, and it is made of cellulose, hemicellulose, and glucoproteins, and another component of pectin. And finally, secondary walls grow from inside the primary walls, and they don't replace them, but they strengthen them by adding lignin, which kind of acts like rebar, whereas the other elements kind of act like concrete. So it's making it really strong and rigid. Communication occurs through cell walls via structures called plasmodesmata, and they go through different pores of the cell wall, which contain tiny strands of cytoplasm which connects to other cells, and then molecules can be passed through different cells and they can communicate that way. The plasma membrane then lies right inside the cell wall, and as discussed before, it is made of a phospholipid bilayer. The phosphorus heads are the little white balls represented there, and then the lipid tails point towards each other, and making this bilayer, which is hydrophilic on the outside and inside, and on the in between the lipid tails is a hydrophobic section. And this allows the controlled movement of substances in and out of the cell. Small things like oxygen ions, other things can generally float through the cell membrane without the aid of other proteins. It also helps to aid in the production of cellulose for the cell wall. But major contributors also to what goes in and out include proteins that are embedded within or across the wall. They may also act as receptors for hormones, may be used to identify different types of cells, and are thus important for the function of the cell. Now, the major kind of powerhouse of the cell, well, this isn't the powerhouse, this is really where the blueprints are kept, kind of the mastermind or the brain of the cell. If anything needs to be done, it has to first be drawn up in the DNA first, which in the nucleus, which contains the DNA, and clusters of DNA are called chromosomes. This is also where replication occurs. So if you want to make two cells, first you have to double the DNA, and that occurs through the process of replication. Transcription also occurs here, where you take a strand of DNA, you unzip it, you make a messenger RNA, and then you transport that messenger RNA out of the nucleus so it can be made into a protein. It exits through the nuclear envelope through what's called nuclear pores. And also you have the dark area usually in the center of the nucleus called the nucleolus. This is where you have a lot of replication or transcription occurring. So a lot of things, enzymes and structural components are found there, which are replicating the cells or making this messenger RNA. Some of the messenger RNA may go to a place called the endoplasmic reticulum, depending on whether there are ribosomes there or not. But the endoplasmic reticulum has a lot of functions, including membrane synthesis, so it'll make membrane for different types of organelles, or maybe even for the plasma membrane itself. It also will modify proteins as they pass through. They may be folded differently or have different tags put on them and so forth. Rough endoplasmic reticulum then is the endoplasmic reticulum that has ribosomes on it. Smooth does not. Ribosomes are actually made of RNA as well. They're made of a ribosomal RNA, or rRNA. It has two subunits. You kind of see them illustrated in this figure. We have a red, which is a larger subunit, and a blue, smaller subunit. They act together with a messenger RNA and transfer RNA to make proteins in a process called translation. The Golgi apparatus has functions kind of similar to the endoplasmic reticulum. It creates vesicles for the transport of certain substances, or may also modify carbohydrates. It can assemble polysaccharides. It can also make sections of the membrane that can be used for vesicles for the plasma or the plasma membrane. So we'll talk about what a vesicle is in a minute. Plastids, so this is the general name for organelles that have photosynthesis, have chlorophyll and contain photosynthesis. But the most abundant one, there are a bunch of different types, but the most abundant one is called chloroplasts. This is shaped like two frisbees that are kind of stuck on top of each other in the same configuration. And so it has two membranes. Within there it has grana, which looks like a bunch of stacks of coins. Those individual coins are called thylakoids, and they contain the chlorophyll and other pigments which are important for photosynthesis. It also has a fluid component called stroma, which has all the necessary enzymes and parent material for photosynthesis. Sorry, my phone keeps on going off. My kids keep calling me and all that stuff. Alright, so this is what it looks like. You have a thylakoid, them all stacked together make a granum. Those all granum all combined are found within the chloroplast. Mitochondria are similar, almost in shape as well, except rather than having stacks, they have an inner membrane that is folded. And these folded membrane make projections, which are called crista or cristae. And this is where cellular respiration takes place. So photosynthesis occurs in the chloroplast, and then respiration takes place in the mitochondria. And so this is the powerhouse of the cell. This is what is making all the, or converting all the glucose and molecules into ATP. So both chloroplasts and mitochondria have their own DNA and actually replicate separately to the overall cell itself. So these both point to the evidence of what's called the endosymbiotic theory, which is that both mitochondria and chloroplasts were once their own free-living organisms that were somehow engulfed by a larger organism, and they initially tolerated each other, but eventually built up this mutualistic relationship where they each can now not live without each other. And they both benefit by making energy and providing materials for that energy. There may also be smaller things called microbodies, which are specialized enzyme in a singular membrane, and these are often called vesicles as well. So peroxisomes contain enzymes for breaking down hydrogen peroxide, which can destroy other molecules that are important, so it can destroy tissues and basically act like a poison kind of to the cell. Gloxosomes contain enzymes that help convert fats to carbohydrates. Lysosomes contain enzymes that help break down, old organelles or maybe a little pathogen that has somehow made it into the cell. Vacuoles are basically, they just kind of look like big bladders. They're full of water and liquid, and also have other important things within them storage-wise. May have sugar or materials for like amino acids and other things. They help maintain the cell pressure, so they help maintain the shape of the cell. They may also help maintain the pH, metabolites, and waste products. They also often contain pigments that give flowers their colors. Finally, you have like an internal structure within the cell to help move substances along, to kind of act like railroad tracks, and that's part of the cytoskeleton. Also to give it structure to kind of, just hold it up and keep it from folding in on itself. The cytoskeleton is made of microtubules and microfilaments. Microtubules are composed of proteins called tubulins, and so you have on this figure a bunch of, these are actually microfilaments, which also play a role in cytoplasmic streaming or cyclosis, where that cytoplasm circulates through the different parts of the cell to deliver things where it needs to go. Alright, so that was the lecture for Friday. This is going to be the lecture for Monday, which goes over the cell cycle, or the different parts, so different stages of the cell as it goes through its different functions and then needs to replace itself. So the cell cycle can be split into two major phases: mitosis, which involves the splitting of the cell into two separate cells, and interphase, which is really the functional part of the cell. So this is where it's doing its job, and then also kind of prepares for mitosis and the other phases. The three parts of Anaphase are the G1, S, and G2 phase. G1 is the first growth phase, and this is where it grows, matures, and fills out its function. The S phase is the synthesis phase, and this is where DNA is being replicated and doubled. It's being prepared for mitosis. In G2, or the growth phase 2, you have the doubling of organelles and prep for mitosis. So now that you have two of everything and everything ready to go, you can now split into two. This occurs through the mitotic phase, the M phase, which is again broken down into 4 different phases. And the acronym you can use is called PMAT. So prophase, metaphase, anaphase, and telophase. During prophase, you have the chromosomes when they're being used, they're all spread out and you can't differentiate. It just looks like the nucleus just looks like a mass. But during prophase, they start to condense, connect at the centromeres. You can see these light and dark areas. And what's happening is the spindle apparatus is assembling, which is going to attach to the chromosomes and split them. The two centrioles move to opposite poles, forming the spindle apparatus. And then the kinetochore is the part of the chromosome that attaches to the centromere. The nuclear envelope then also breaks down because you're going to have to separate and make two new nuclear envelopes. This should be a 2, but during metaphase, the alignment of the chromosomes occurs in the middle of the cell, and that is a place called the metaphase plate. The spindle fibers align them there. But the metaphase plate is not an actual structure, it is just kind of an imaginary line where they line up, but it eventually will become where the cell divides. Anaphase is the third, not fourth stage, and this begins when centromeres split, so the chromosomes split. And the key event is the removal of cohesion proteins, which are holding them together, from all the chromosomes, and now they are separating and moving to opposite ends of the cell. And you get one of each, and those sister chromatids then, which were originally on one chromosome, are now split into two. Telophase is then the fourth phase, and this is where the spindle apparatus is no longer needed. It disassembles and is no longer attached to the chromosomes. The nuclear envelope reforms. You get two of them. And now you have your chromosomes unraveling and start making their start completing their functions. The chromosomes also then begin to uncoil, and the nucleolus reappears as the activity occurs in the DNA. The final part is called cytokinesis, and this is the splitting of the cell, or the cleavage of the cell into equal halves. And then in plant cells, you have a cell plate that must form to complete the cell wall between the two cells.